Hello everyone, and welcome to today's webinar, Endpoint Assays for the Characterization of Rare and Therapy-Resistant Cells in Cancer. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. This presentation is educational and thus offers continuing education credits. Please follow the process to obtain your credits by clicking on the Continuing Education Credits window. I'd like to now welcome our speaker, Jordi Petriz, Group Leader, Functional Cytomics Group, Josep Carreras Leukemia Research Institute in Barcelona, Spain. Jordi, you may now begin your presentation.
Okay, thank you very much for this nice introduction. First of all, I would like to thank the organizers for inviting me and giving me the opportunity to explain how the endpoint assays that we are developing at the Josep Carreras Leukemia Research Institute are crucial for the characterization of rare and therapy-resistant cells in cancer. In general, a typical experiment involves four basic steps, such as the adequate sample preparation, the optimized instrument setup, the procedure of data acquisition, and finally, how we perform data analysis. We are particularly interested in human leukemia, and we are currently developing new methods to both identify healthy and leukemic stem cells. Stem cells are rare, and the accurate identification of this compartment is crucial for human progenitor cell counting and for monitoring minimal measurable disease, among other practical stem cell assays. More than 20 years ago, we published this article in Nature Medicine as a new technology to identify CD34 positive cells in analyzed whole blood. By using viable nucleic acid stains, we avoided red cell lysing, a procedure that can result in leukocyte depletion as well as detrimental for CD34 positive cells. As you can see here, the first plot is used to display CD45 positive nucleated cells, simultaneously stained with cytosine, a permanent nucleic acid dye used to exclude non-nucleated cells. A fluorescence trigger was used to discriminate leukocytes from erythrocytes, and after a sequential Boolean gate, CD34 positive cells were enumerated using the last plot. Counting bits are also displayed here and use it for absolute CD34 cell counting. No lyse, no wash methods are deployed when minimal sample perturbation is needed. Cells are directly stained and diluted in Higgs valent cell solution with no detrimental viability and functional effect. Then, analyzed blood can be analyzed after dilution up to 10-fold faster speed rate using acoustic assisted flow cytometry. Leukocytes are, in general, outnumbered by red blood cells in approximately 1,000-fold in whole blood, and generally require enrichment by red blood cell lysis or gradient centrifugation prior to analysis. The rapid sample collection rates and inclusion of the Attune NXT flow cytometer allow identification of leukocytes by scatter properties alone when using, for instance, the violet side scatter laser. Leukocytes, even diluted rare cells, can be easily identified using no lyse, no wash methods. By using the violet laser configuration and a trigger for CD45 fluorescence, red blood cells are completely discriminated. CD34 positive cells can be counted as demonstrated here, providing highly accurate and precise enumeration. As shown here, even using high-speed sample rate, less than 3 CD34 cells per microliter can be detected. In this slide, we have compared identification of CD34 leukemic cells. On the left, cells were analyzed using a conventional hydrodynamic flow cytometer and single laser excitation. On the right, the same specimen was analyzed using our no lyse, no wash approach with two laser excitation to better excite phycoerythrin, the dye we commonly use for CD34 conjugates. As you can see here, there is a very good separation of leukemic cells suggesting clonal evolution of the leukemic population using the acoustic cytometer and this no lyse, no wash approach. In this slide, we can see a flow cytometry analysis of analyzed cord blood, as we shown here. A threshold is set for determining nucleic acid labeling whole blood live cells by cyto13 versus PC5 CD45 analysis of DNA RNA positive events in R1. On the right, forward scatter versus CD45 negative events are shown in R2, and CD45 negative population are mainly nucleated red blood cells, as is shown here. The scatter properties of leukocytes and nucleated red blood cells are also shown here, and on the right, a new scatter plot is used to show a range of immature to mature nucleated red blood cells. Extruded nuclei are identified as a very low forward scatter population, and low forward events mainly contain orthochromatic erythroblast, and nucleated red blood cells with blast scatter pattern are early stage erythroblasts. Cyto13 positive CD45 negative population were then sorted on the basis of forward scatter properties and prepared for microscopy analysis. The morphology of this series was in agreement with erythroid differentiation from early to late stage erythroid tests, with a decrease in cell size. And I would like to show you now how acoustic focusing takes advantage for many applications. And particle focusing is promising. As an example, particles can be oriented easily by simply using specific frequencies. The particles that we are observing here, and after increasing the frequency, and as you can see here, particles can be specifically oriented based on the frequency that we are applying. And in general, higher frequencies will provide more complex patterns, as also shown in the present and the two next slides. Adding more complexity at the pattern of distribution of dust inside over this plate, and after increasing the frequency of the waves that are producing this vibration in this magnetic base. The field of acoustic cytometry was originally invented at the Los Alamos National Laboratory as a way to allow scientists to quantitate and examine cells by passing them through a laser-based detection device. Acoustic focusing enables both longer transit times and high throughput, which simultaneously permit better interrogation of every cell in a sample, and analysis of much larger number of cells, bringing researchers and scientists one step closer to the precise quantification of molecular phenotypes at the single cell level. The following two slides show how, when the acoustic focusing is turned off, microbeads are misaligned across the capillary section. And as you can see here, the beads are misaligned with the acoustic focusing off. And however, when acoustic focusing is turned on, particles are clearly aligned, allowing optimal laser excitation even at higher sample rates. Therefore, coincidence of particles will be minimized when optimal cell or particle dilutions are used, taking advantage for rapid and sensitive detection of rare cells. Laura Garcia Rico, a postdoc working in my lab since 2015, conducted most, if not all, the experiments I will start to explain right now. As you may know, like the scatter patterns generated from a cell size and complexity can be used to broadly identify different cell types, consisting predominantly of lymphocytes, monocytes, and granulocytes when we are looking at blood. Several techniques have employed electron microscopy to the study of blood cells, scanning microscopy in the upper row and transmission microscopy below provide differential information, being the last one specifically used to demonstrate chromatin changes and condensation. In this slide, a representative scatter plot of normal whole blood is shown, and as you may know, forward scatter provides information on the relative size of the analyzed events, while SAT scatter estimates the complexity. In general, monocytes are larger than lymphocytes, while neutrophils have more complexity or granularity. However, the observed classic profile results from the overlap of different subsets of leukocytes with different light scatter properties that can easily identify it on the acoustic flow cytometer when appropriately analyzed. Now, with the aim of identifying primitive leukemic progenitor cells in human leukemia, we modified an alkaline phosphatase stem cell detection method based on the determination of a fluorescent alkaline phosphatase substrate in cells positive for the enzyme. The original method, using fluorescence microscopy, was adapted for flow cytometry measurements, and alkaline phosphatase live stain is the proof that we have used it for this kind of experiments. Is a cell-permeable fluorescent substrate for alkaline phosphatase and that is non-toxic to cells and diffuse over the cells after the course of many hours. We have used this method to identify different subsets of CD34 positive cells based on the activity of the enzyme. And as you can see here, mobilized peripheral blood and the leukapheresis products were analyzed, showing representative plots as we shown here. And we hypothesized that CD34 positive cells with high levels of alkaline phosphatase activity are the more primitive progenitors, and that can be used as a potential biomarker for short and long-term engraftment in human transplantation. After stem cell mobilization, absolute counting of CD34 positive cells was compared, and healthy donors and multiple myeloma patients showed different alkaline phosphatase activity, as shown in the next slide. We also believe that CD34 positive alkaline phosphatase high cells may be associated with a more primitive stem cell-like population, being essential for long-term engraftment, whereas CD34 alkaline phosphatase positive cells with low activity of the enzyme may be associated with more differentiated populations with short-term repopulating capacity. Importantly, we are optimizing CD34 positive cell counting methods, and we are currently applying specific improvements aimed at the more precise progenitor cell counting. The NXT cytometer is ready to provide true direct volumetric counting without bits. In addition, PC34 positive cells can be detected after excitation with a yellow-green laser, giving optimal separation from negative cells. This slide shows how we usually discriminate doublets and aggregates once the rare fraction of cells has been identified. Based on the use of nucleic acid stains for no-lyse no-wash methods, better resolution for single-cell discrimination is obtained when a fluorescent parameter is used. The next following slides will show you how sample manipulation heavily affects a special subpopulation of fragile cells, which are also rare. Dendritic cells and basophils can be identified as described in this slide, and plasmacytoid dendritic cells and conventional dendritic cells can be discriminated based on the positivity and negativity of CD123 and CD11C markers. This slide shows the results we have obtained using the no lyse no wash approach, and a dump channel was used to discriminate lineage negative cells and then plotted again to both identify basophils and the two types of dendritic cells. This was done using the no lyse no wash method, and this reference method was compared with different lysing solutions such as Cy-Lyse, OptiLyse solution, and Pharmalyse, a non-fixative ammonium chloride based solution. And as you can see here, there are important differences in the resolution and the scatter properties of the populations that we are analyzing. And just to show you how this slide summarizes the four methods we have compared, showing representative plots for dendritic cell counting. Values displayed here represent the absolute numbers of dendritic cells obtained for comparison using the no lyse no wash method, the Cy-Lyse, the OptiLyse, and the ammonium chloride lysing solution. We then analyzed and compared the numbers of conventional dendritic cells, the number of plasmacytoid dendritic cells, and basophils, showing that red cell lysing is heavily affecting the absolute counting of these rare cells. As a take-home message for this part of my talk, I would like to conclude that red cell lysing methods should be avoided if possible, making feasible the detection of rare cells even using diluted and unlysed whole blood specimens. Many of the experiments performed in our laboratory are functional based assays. After sample reception, blood cells are immediately prepared for flow cytometry analysis in as little time as possible. Over a period of 1 year, we have been using this technique to study its activity in patients with leukemia and lymphoma, showing that changes in the alkaline phosphatase levels can be used to detect rare population of highly refractory malignant cells. Here is shown 1 year history from my follow-up of one patient. We published these results in this article in 2016. And this patient had 27 years old, was a female, diagnosed in March 2010 with common acute lymphoblastic leukemia with normal karyotype. Antigen expression was evaluated at first relapse, showing 40% of CD34 blast cells with identical phenotype of refractory disease as compared with diagnosis. Red circles shown here indicate the bone marrow flow cytometric analysis to detect alkaline phosphatase activity in combination with CD34 positive staining. The alkaline phosphatase test was adapted from previously described methods and only was used to determine the alkaline phosphatase activity when was internally validated. This slide is and the following will show you how the presence of apparently clonal populations does not necessarily represent clonal neoplasia. Upper and lower panels display comparative phenotypes at diagnosis and first relapse, respectively, whereas this slide is used to display comparative phenotype at second and third relapse, respectively. As shown here, a phenotype of apparently clonal leukemic cells was based on CD10, CD19, CD33, CD34, and CD66 positive expression. One possible explanation for the amplification of the apparently monoclonal leukemic cell is clonal evolution, and pre-existing resistant subclones could also explain cases arising from very rare premalignant hematopoietic cells, constituting a potential cellular reservoir that may play a key role in continuously seeding relapsed disease. The alkaline phosphatase assay was internally validated and was used over the last year of treatment of this patient. 5 bone marrow aspirate specimens were taken from May 2015 to April 2016. And by using the alkaline phosphatase assay, our results suggest that the apparent clonal leukemic refractory cells can be classified into functional states based upon the different activity levels of the phosphatase. If true, this method could help to identify and to differentiate new oligoclonal or pseudoclonal populations in patients with neoplastic malignancies. This slide also shows the performance of the alkaline phosphatase test in combination with CD34 staining in a second case that we also studied with CD34 positive alkaline high cells at diagnosis and relapse. Each individual circle represents CD34 positive cells with high expression levels of alkaline phosphatase activity, presumably enriched in primitive refractory cells. Comparisons of well-defined encircled populations consisting of these CD34 alkaline high cells provides valuable information at the moment of diagnosis and similarly at relapse 6 months later. In order to evaluate the potential role of alkaline phosphatase positive cells in lymphoma, we applied our methodology to a 12-year-old patient with peripheral blood immunophenotyping compatible with the diagnosis of Burkitt's lymphoma, together with a bone marrow or spheroid infiltration, pleural effusion, and ascites, with 15 to 87% of CD10 positive, CD19 positive cells. Flow cytometry analysis of CD10 positive, CD19 positive cells and alkaline high positive lymphoma cells were found in marrow as shown here, and also evidencing an increased activity of alkaline phosphatase positive cells in ascites lymphoma cells that we also identified. Because stem cells lack specific cell surface markers, the identification of this compartment can be difficult, making cancer stem cells especially elusive. Thus, we undertook a prospective cohort study based on our previous work, with the objective to investigate whether there might be differences in alkaline phosphatase activity within primitive leukemic cells and its association with the potential risk of recurrence and mortality in newly diagnosed patients with acute myeloid leukemia. Between 2015 and 2018, a total of 106 patients were diagnosed with acute myeloid leukemia in our hospital, and after exclusion criteria that we applied, 43 eligible patients were identified and included in the overall analysis. Briefly, our assaying strategy used a dicycle violet stain, a low cytotoxicity permeant DNA-specific dye that can be used for DNA content cell cycle flow cytometry, but also to identify very primitive stem cell site, like the site population. Dicycle violet can be excited with violet laser light, and can be used for simultaneous measurement with alkaline phosphatase, which is excited with blue laser, and its emission can be collected using a standard fluorescent filter. Subsequently, cells were stained with fluorophore conjugated monoclonal antibodies and can be easily analyzed on the flow cytometer. In this slide, more detail is provided to better understand how we discriminate alkaline phosphatase positive cells. Side scatter was detected using the violet laser, and alkaline high positive cells were simultaneously detected with monoclonal antibodies that we selected to better classify the leukemic stem cell compartment. The CD34 positive cells, this is phycoerythrin CD34 positive cells, were detected after excitation with the yellow-green laser, and the alkaline phosphatase light stain was excited again with a blue laser. The improved signal, together with the fact that there is no need for color compensation, results in the improved immunophenotyping analysis of leukemic cells by multicolor flow cytometry. Two bone marrow aspirates are compared in the same patient, displaying high levels of alkaline phosphatase activity in combination with C therapy for the staining and diagnosis and relapse. And we used a receiver operating characteristic analysis, area under the curve, sensitivity and specificity to measure prognostic accuracy of our test in predicting event-free survival, and the optimal cutoff value was identified according to the Jordan's index to classify patients into two groups according to the 12% of alkaline phosphatase life-stain positive cells within the blast population that we detected using the previous panel I was showing in the previous slide. Univariate and multivariate analysis were used to identify factors that are associated with overall survival and event-free survival in patients with acute myeloid leukemia. Alkaline phosphatase expression was a significant predictor of overall survival on univariate analysis. Alkaline phosphatase expression was a significant predictor of event-free survival on univariate and multivariate analysis. And in the univariate Cox model for overall survival, age, favorable and intermediate risk, AML type, and alkaline phosphatase group were associated with a significant impact of survival. The only factor that retained their significance on multivariate analysis was age, and in the univariate Cox model for event-free survival, age, favorable and intermediate risk, and the alkaline phosphatase group were associated with a significant impact on progression and relapse. The only factors that retained their significance on multivariate analysis were favorable risk and the alkaline phosphatase group. And plots of Kaplan-Meier lineage estimates of overall survival of acute myeloid leukemia and group patients are shown here, and ROC curve allowed us to classify two identifiable alkaline phosphatase groups of patients at diagnosis according to the numbers of leukemic blasts expressing more than 12 or less than 12% of the alkaline phosphatase positive cells. These are 27 patients in group A, and less than 12% of alkaline phosphatase positive cells, 16 patients in group B. As well as for the event-free survival when classified by the alkaline phosphatase group, as can you see here. Cancer immunotherapy was named the Science by Science magazine as the breakthrough of the year. And more recently, the Nobel Prize for Medicine and Physiology was awarded to Jim Allison and the Suku Onjo in 2018. And cancer immunotherapy will cover the last part of my talk today. In order to advance personalized medicine in oncology, PDL testing for anti-PD-L1 directed immunotherapy has emerged as a highly promising biomarker. PD-L1 is one of the ligands for PD-1 that can be expressed by tumor cells and may also be induced. PD-L1 tumors have been shown to be more likely to respond to checkpoint blockade. However, complexities exist because too many antibodies and cutoffs use it for PD-L1 and is not a perfectly predictive biomarker at the present time. Checkpoint antibody inhibitors are a novel class of inhibitors that function as a tumor suppressing factor via modulation of immune cell-tumor cell interaction. These checkpoint blockers are rapidly becoming a highly promising cancer therapeutic approach that yields remarkable anti-tumor responses with limited side effects. And despite the huge success and efficacy of the anti-PD therapy response, it is limited to specific types of cancer, which attributes to the insufficient and heterogeneous expression of PD-1 in the tumor microenvironment. 3 years ago, I had the opportunity to attend this very exciting meeting, this scientific tutorial focused on antibody validation for flow cytometry, and many concerns were raised about PD-L1 reactivity by the organizer, but also by the attendants. The prognostic value of PD-L1 has been controversial in recent studies, and PD-L1 is known to play a major role in suppressing the immune response. Yet increasing studies have reported that PD-L1 expression has a favorable prognostic value for cancer patients. This raises the concern about how to understand PD-L1 expression, and this table highlights the importance of PD-L1 affinity, and it suggests that the reactive efficiency of PD-L1 is more useful to predict the response to immunotherapy. Then we aimed to determine whether PD-L1 expression could predict the prognosis of newly diagnosed multiple myeloma and lung cancer patients, and we decided to develop a highly reproducible for accurate scoring of PD-L1 positive cells. We have validated a in-house cytomic assay that can be performed on peripheral blood and bone marrow samples at diagnosis, and this test can be used as a guide to predict response to anti-PD-L1 directed therapies to target the PD-L1 pathway with therapeutic antibodies directed at PD-1, because disrupting the PD-L1 pathway with therapeutic antibodies results in restoration of effector immune responses against the tumor. Because tiny biopsies can result in false negative PD-L1 evaluation, we focus on the accurate identification of PD-L1 on peripheral blood myeloid-derived suppressor cells using flow cytometry and our no lyse no wash methods. And as you can see here, we can detect the target using a specific antibody against PD-L1, and then register the fluorescence and collect all the events on the flow cytometer. Recent studies have shown that the presence of myeloid-derived suppressor cells in multiple myeloma bone marrow microenvironment contributes to local suppression of the immune responses through the PD-1 pathway. These subsets constitute a heterogeneous population that expands during cancer, inflammation, and infection, playing a critical role in the progression of multiple myeloma. And there is a growing interest in this population as a therapeutic target for this disease. The use of the anti-PD-1, PD-L1 monoclonal antibodies in cancer patients might represent a novel therapeutic strategy, and this study was aimed to develop novel strategies to detect PD-L1 expression in myeloid-derived suppressor cells. Another controversial issue that was apparent from the beginning is how to identify these cells. The field of myeloid-derived suppressor cells research is still immature, and it's difficult to distinguish these cells from neutrophils and monocytes. And this slide shows mice and human phenotypes, but the exact function of myeloid-derived suppressor cells remains still controversial. Myeloid-derived suppressor cells are increased in multiple myeloma and lung cancer, express CD33, low or negative HLA-DR, are CD11B positive and PD-L1 positive. And we also decided to use a scheme of 4 laser-like excitation to minimize color compensation to provide the optimal screening of multiple myeloma myeloid-derived suppressor cells, because PD-L1 expression may help to identify patients most likely to respond to PD-1 PD-L pathway inhibition alone or plus inhibition of another pathway. Here is displayed a representative no lyse no wash protocol for the screening of PD-L1 myeloid-derived suppressor cells by combining dicycle violet to discriminate nucleated from non-nucleated cells and a series of markers to finally identify monocytic and polymorphonuclear myeloid-derived suppressor cells. Evaluation of PD-L1 expression was performed in terms of cell numbers and antigenic density comparisons across stimulation. And as you can see here, after stimulation of PD-L1 positive cells were undetectable in this patient, and changes in PD-L1 expression after 4-Ester treatment with PMA found a series of patients with a dramatic increase after stimulation, compared with non-stimulated cells, suggesting that fold change in PD-L1 appears to have an important regulatory factor that may relate to difference in underlying mechanisms within patients. Importantly, PD-L1 was not found at cytoplasmic level. Variation in PD-L1 fold change among patients led us to consider the possibility that PD-L1 is expressed at cytoplasmic level and after stimulation translocated to the cell membrane in the same manner as CD11b. PD-L1 and CD11b cytoplasmic levels were simultaneously studied in 11 patients, and PD-L1 was found to be undetectable at cytoplasmic level in comparison with CD11b also at cytoplasmic level for reactive antigen. And this slide shows the PD-L1 fold change in a series of 31 multiple myeloma patients. Fold change was calculated as a ratio of number of stimulated and non-stimulated myeloid-derived suppressor cells PD-L1 positive, and all patients showed a wide fold change variation ranging from 1, which means no variation, to 650, which means a large variation. In this slide, we can visualize the molecular mechanism of interaction between PD-1 and PD-L1 and blockade via durvalumab to the tiny accessible area of less than 2 angstroms. Competitive experiments in presence of durvalumab were also performed in my lab, and the PD-L1 conjugate for flow cytometry analysis confirmed near identical target site, as we demonstrated with this kind of competitive experiments. PD-L1 is detected differentially depending on the stimulation time, and the fact that the PD-L1 was not present at cytoplasmic level led us to investigate changes in PD-L1 expression over time, and after stimulation, PD-L1 expression was found to be higher after 1 to 5 minutes with a progressive decrease up to an hour. Okay. Oops. And this graph was used to display the PD-L1 kinetics in terms of mean fluorescence intensity and percentage of PD-L1 positive cells. PSNE visualization methods helped us to monitor that other phenotypic changes did not occur with the stimulation over time, whereas PD-L1 was the only marker affected over time, as you can see here. And the following slides will show you how PD-L1 is quickly changing to the unfolded state after stimulation. You can see here, this is 1 minute, this is 5 minutes, 4 minutes, up to the final slide completed at 60 minutes. Finally, I would like to highlight our more recent publication demonstrating how PD-L1 reactivity appears to result from complex interactions that can only be detected with minimal sample perturbation and using these no lyse no wash methods. In view of these conformational properties, analysis of pathological immunosuppressive myeloid-derived suppressor cells could help to better understand and overcome the mechanisms of immunotherapy resistance by developing new treatment strategies aimed at promoting PD-L1 stabilized and folded states. Altogether, our findings may indicate the need to find compounds that by themselves exhibit a PMA-like effect aimed at abrogating this pathologically expanded myeloid-derived suppressor cell population and not only killing the tumor cells. In this slide, a more classic display was used to monitor differential PD-L1 expression before day 0 and after immunotherapy, suggesting that the unfolded state can be a novel mechanism of resistance to cancer immunotherapy, as well as to show how immunotherapy has little effect on the numbers of pathologically expanded myeloid-derived suppressor cells. In summary, panels of immunomarkers in combination with functional analysis can provide more information than single phenotype. Our protocols allow untouched processing of whole blood and bone marrow by simply adding reagent or alternatively by collecting violet versus blue side scatter and intrinsic cellular alkaline phosphatase activity diagnosis may be used to estimate relapses and disease persistence more accurately. Our immunotherapy study is aimed to develop novel strategies to detect PD-L1 expression in both myeloid-derived suppressor cells to predict anti-PD-L1 therapy success. And this PD-L1 myeloid-derived suppressor cells, which are increased in cancer patients, having important roles in these immune suppressive functions that we will need to clarify and control by decreasing this number circulating in peripheral blood. Finally, I would like to thank my team, Angel Bistuel, Laura García Rico, Jorge Bardina, Jordi Junca, Marc Suriguer, and José Salvia. And I will like, and very especially, I would like to thank Jolene Bradford and Michael Ward for providing the support which allowed me to undertake this research and for the professional and personal relationship over the last 9 years. Thank you very much for your attention, and I will be happy to take some questions from the audience.
Thank you, Jordi, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is PD-L1 expression a biomarker for patient selection? Is PD-L1 really a predictive biomarker?
Oh, thank you very much for this interesting question. I would like to say that we are exploring. PD-L1 expression can be a biomarker for patient selection. We are currently investigating some lung cancer patients. We have started a prospective study, and we are exploring the PD-L1 expression using this no lies, no wash method, this functional analysis in my lab, just to try to determine if the PD-L1, this change in the folding state, can be really a predictive biomarker. One of the problems with tumor cells is that in many occasions we can receive a tiny biopsy, but we prefer to use this non-invasive method just to study the PD-L1 expression on this expanded, on this pathologically expanded myeloid derived supercell cells. And I have no preliminary data, but I think that we will be able to obtain very nice information, not also related as a potential biomarker for patient selection, but also for try to understand if specific or some mechanisms of immunoresistance can appear.
Thank you, Jordi. Our next question is there any physiological function for alkaline phosphatase in normal stem cells?
Whoa, this is also a very nice question. My suspicion is that the alkaline phosphatase plays in the alkaline phosphatase activity, or this enzyme plays a key relevant role in the bone marrow microenvironment. I think that I have no demonstration of this hypothesis, but I believe that the alkaline phosphatase in normal stem cells is contributing to develop a more hypoxic niche, which is preferred for this kind of for the more primitive healthy stem cells, and just to avoid a critical differentiation to non-desired lineages, and just to preserve the self-renewal capability.
Okay, great. Thank you so much, Jordi. Our next question is PD-L1 involved in all human malignancies?
Well, we have this is very well described, and it was I was explaining that in one of my slides that this the PD-L1 expression is not related or is not expressed in all tumors. It can be expressed in solid tumors, but also in leukemia. And we are especially focused in the beginning. We were especially focused in multiple myeloma, but immunotherapy was discontinued, and that's the main reason, because we are now studying the PD-L1 expression in myeloid derived supercell cells from patients with lung cancer. But it can be expanded to other cancers, but always limited by the PD-L1 expression in tumor cells. I think that it is necessary to highlight that PD-L1 can be also expressed in the myeloid derived supercell cells, which can be expanded in many human malignancies, contributing to tumor proliferation. And maybe this can be a very hot topic, not to only study the expression of PD-L1 in tumor cells, but also studying the expression of PD-L1 in this pathologically expanded population.
Thank you, Jordi. Looks like we have time for a few more questions. Our next question here is alkaline phosphatase the only available technique to detect cancer cells?
The answer is clear. The answer is no. There are many methods that were designed to identify cancer stem cells. One of the methods that we prefer in my lab is not only the alkaline phosphatase expression. We also look at the expression of multidrug transporters. And a different question is if this technique can be also used to detect cancer stem cells. Of course, this technique, the alkaline phosphatase, is available as others, such as the aldofluor and other methods to detect cancer stem cells. And this alkaline phosphatase method takes advantage to other methods that are available in the market, because it's not effluxed by multidrug transporters. which are overexpressed in cancer, can be overexpressed in cancer cells, but also in cancer stem cells, such as the cytopopulation with overexpression of the ABCG2 transporter.
Thank you, Jordi. Our last question is the alkaline phosphatase method necessary or enough for a cancer stem cell phenotype? How is alkaline phosphatase positivity defined?
Yeah, I will start by the second question, because we were reanalyzing the values that we obtained in our cohort of 43 patients with acute myeloid leukemia, and we can use the ratio between positive and negative blastic cells by comparing the alkaline phosphatase activity, and not only the 12%. I think it works very well when comparison with the cutoff that we obtain using the ROC curve analysis. The ratio between alkaline high and alkaline low positive cells appears to be a reliable method to discriminate this positivity within the leukemic stem cell population. And related to the first question, if alkaline phosphatase method is necessary or not for a cancer stem cell phenotype, I think that this method can be complementary. And one of the main advantages of using the alkaline phosphatase staining is that we are detecting a very rare fraction that cannot be identified by using a phenotype, but we can combine this fluorescence coming from the activity of the enzyme with many other antibodies, but also with other fluorescent proofs to better define the cancer stem cell population. And this takes advantage because we can isolate by cell sorting these populations, and then perform single cell sequencing, and use study gene expression analysis of the cells that we are isolating. And this, I think that this can be a very nice tool that in the future research will need to clarify how these cancer stem cells are expressing this enzyme at higher level. There is a approach that we also developed in my lab, because we were studying astrocytomas and gliomas, and we were interested in identifying the site population. And we observed that under hypoxic conditions, the number of site population cells increased, but also increased the number of alkaline phosphatase positive cells, or alkaline phosphatase high cells within the site population, indicating that we are enriching in more primitive site population cells, not only by the classical multidrug resistance efflux of the HDI or dicycle violet, but also confirmed by this increase in the alkaline phosphatase activity.
Thank you again, Jordi, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.